Welcome to this session in biology. In this session, we'll be looking at the topic genetics. At the end of this lesson, you will be able to define and explain heredity, explain chromosome and transmission of characters, and also explain sex-linked characters. Join me as we go through this lesson. Now you'll be wondering and asking questions. How are characters transmitted from your parents to you? If your dad has an oblong head, how come you also have an oblong head? If your dad has the characteristics of intelligence, how come you also have the characteristics of intelligence? If your mom has certain characteristics that you have, how did you get those characteristics? That is basically what we're looking at. And that's under the study known as heredity. Heredity, this is the transmission of characters from parents to offspring. That's what heredity entails. And we'll be looking at different types of hereditary characters. So heritable characters are: 1, body structure and shape of head, nose, and ear. We also have blood grouping, ability to taste PTC, phenylthiocarbamide. It's usually very bitter in some people's mouth, but in some others, they don't feel that bitterness or sourness. Then we also have characteristics of voice or speech. We have fingerprints. We have intelligence. And we have hemophilia. It's also a heritable characters. We have a whole lot of heritable characters. This is just to mention a few. Now, our study is based on the works of a scientist known as Gregor Mendel. And he lived between the year 1822 to 1884. He carried out his experiments on pea plants. The reason why he carried out his experiments on pea plants, which is also known as Pisum sativum, is because of these various characteristics. The seeds were round. Some seeds were round, whereas some seeds were wrinkled. Some of them had green pods, whereas some had yellow pods. Some seeds were yellow, some seeds were green. Some flowers were purple, some flowers were white. I'm talking in terms of the petals. Then some pods were inflated, whereas some pods were pitched. Then he had terminal and axial buds. That means some plants had terminal and axial flowers. They had terminal and axial flowers. Some of them had long and short stems. So those are the various characteristics that made Gregor Mendel to also choose them. Another significant point is that they are annual plants. He can get his results within a limited time. That's why he worked on these plants. They are annual plants. All right. We are also going to look at other types of laws that this man created. The Mendelian laws. They are of two types. We have the law of segregation. This states that genes are responsible for the development of an organism, and it exists from generation to generation. That means it is passed from generation to generation without being altered or changed. And he also brought up another law known as law of independent assortment. This states that characters exist as a unit, and they are independent of any other character, which I will explain by making various examples. So we are going to look at heritable characters in plants. If you see this is what is known as a Punnett square. This is a Punnett square. And in this Punnett square, we have tall plants and short plant characteristics. The first one you see here, which is this. This is a parent. This individual is a parent. Now, there are certain things that I want you to take note of. If you see a capital T and a short t, it means that this capital T, which is for the characteristics of tallness, dominates the short t. This is a heterozygous tall plant. The reason why it's called heterozygous tall plant is because the genes are different. Dominant and recessive gene for tallness. The capital T is for tallness, the short t is for recessive for shortness. Then we also have another one here. This is another parent. This is another parent. It is also heterozygous for tallness. Capital T for dominance for tallness, and short t, which is recessive for shortness. But the capital T masks the shortness. So it is tall. When we look at it physically, it is tall. So if you make a cross between these two organisms, these are the offsprings that you're going to get. These are the progeny that you're going to get. So you're going to say capital T cross with capital T will give you TT. You also cross this capital T with short t, it will give you capital T short t. You now make a cross of this short t with the first one, capital T. This will give you this. Also make a cross short t with a small t. It will give you this. Now, interpretation of this. The first offspring here that you see is a tall tree because it's made up of homozygous gene for tallness. When you say homozygous, it means that the genes are the same. Then the next one is capital T short t. This is heterozygous for tallness because the capital T masks and dominates the recessive gene for shortness. The same thing applicable here. Whereas in this case, in the case of the last one here, small t, it means that this is recessive for shortness. Recessive for shortness. So that recessive character has really showcased, has really come into play when you talk about this offspring. You can be asked questions like, what is the genotypic ratio? What is the genotypic ratio? When you talk about the genotypic ratio, you're actually talking about the genes that make up these various individuals, So the first one is capital T, capital T. This is homozygous gene for tallness. So that's one, which you have here. The second one is capital T, short t. This is heterozygous gene for tallness, and we have two of them. Hence, the two that you have here. Then the last one is the only one that is available, which is recessive gene for shortness. And that's why we have just one here. So that is the genotypic ratio. Whenever you're asked about genotypic ratio, that is how it's being calculated. Then when you talk about phenotypic ratio, you're talking about the things that you can see, the physical appearances that you can see. If you look at this very well, you'll find out that you have three tall plants. 1, 2, 3. That's why we have 3 here. And then we have one short plant, which is 1. So the phenotypic ratio is 3:1. Now, if you are asked, how many homozygous, what's the percentage of homozygous tall plants that you have here? It's 25% because one, let's say, for instance, these are the different boxes. This represents 25, 25, 25, 25, altogether 100%, right? So we only have one homozygous gene for tallness, that's 25%. So the answer is 25. If you're asked for heterozygous gene for tallness, it is 50 because we have 2, 25 plus 25, that's 50%. If we have for short, we only have 1, and that's 25%. So in the case of phenotypic ratio, if you're asked for in the case of percentage, it is 75% because we have 3, 25 plus 25, 50, 50 plus 25, that's 75. So that's 75%. Then for shortness, we only have 1, and that's 25%. So that's basically how it's being done when you talk about probability and calculation of percentage. The next one we're going to look at in terms of roundness and wrinkledness of a plant. So in this case, the plants could either be round or wrinkled. When the seed is round, it means that it's dominant compared to the wrinkled. Wrinkled is a recessive trait when you talk about the seeds of a plant. So we're going to look at the calculation here. We're going to look at the cross. So capital R plus capital R will give you homozygous dominance for roundness. Then capital R with small letter r will give you heterozygous roundness of seed of plants. Then we have small r with capital R. It will give you capital R and small letter r, which is heterozygous gene for roundness. And then small r with small r will give you recessive gene for wrinkle seeds. So that is basically how the crossing tactics is being carried out. Then we are going to look at chromosomes. Chromosomes is basically the structure that is found in nucleus that carries the DNA, that carries deoxyribonucleic acid. And this is that part that is very much responsible for the transmission of traits from parents to their offspring. This is exactly how it looks like. It's connected by a centromere. The centromere connects the chromosomes together. And each half of the chromosome is known as a chromatid. It's known as chromatid. So in humans, for instance, half of the chromosomes are being given by the parent, each parent, that is the mother and the father. And it totals up to 46 chromosomes, but it has 23 pairs. So 23 pairs of genes, of chromosomes containing genes, are being transmitted from the father and 23 from the mother to make up an individual. We're going to talk exclusively on that. But first and foremost, we're going to look at deoxyribonucleic acid, the DNA structure. The DNA structure comprises the nitrogenous bases. It has a nitrogenous basis. And this nitrogenous basis comprise the adenine, the thymine, the guanine, and the cytosine. Now, these various nitrogenous bases, they complement each other, as in they have affinity for each other. For the case of adenine, adenine has attraction for thymine. For the case of thymine, attraction is for adenine. Guanine has attraction for cytosine. And then cytosine has attraction for guanine. They are all held together by bonds known as hydrogen bonds. Not only that, the DNA also comprises deoxyribose sugar, phosphates. That is what it comprises of. Now, there are certain situations whereby the DNA structure becomes disorganized. When it becomes disorganized, it looks exactly like this. This is when it becomes disorganized. But after a while, it rearranges itself. And that connection is brought about by the hydrogen bond. Adenine connects with thymine, guanine connects with cytosine. RNA has the same type of arrangement, but in the case of RNA basically comprises of uracil. It also has adenine and it also has guanine. Yes, it also has guanine. So that is what the RNA comprises of, and it consists of, and it's known as pyrimidines. Whereas in the case of DNA, it's known as purines. The combination of all those bases is known as purines. So this is the helix structure of the DNA. So that's basically it for the DNA structure. Now we're looking at codominance. Codominance is a bit contrary to what Gregor Mendel postulated. Reason is because Gregor Mendel talked about two laws of segregation, that genes are responsible for an individual and is being transmitted from parents to offspring without being altered, and law of independent assortment, which also states that characters are responsible and they act as a unit in an individuals, and they are independent of any other characters. In the case of codominance, it's contrary to those postulations, those laws. Codominance were carried out by certain scientists, and they say that in the case of codominance, that two dominant genes express themselves together. That is the case of codominance. No one is more advanced or masks the other. They express themselves at the same time, as in the case of checkered chicken. As you can see here in this diagram, this is a checkered chicken that has both colors, black and white. I'm sure many of you have seen such a chicken before. The parents, these are the parents, black parents and a white parents. So let's do the crossing. This is one parent, which is black. This is another parent, which is white. They are both dominant. This is homozygous dominant for black. This is homozygous dominant for white. If you cross them together, the first progeny or offspring is checkered. Checkered, checkered, and checkered. So the F1 generation is all checkered. Now we're going to look at another interesting aspect, which also goes contrary to Mendel's law, which is incomplete dominance. In the case of incomplete dominance, what actually happens is that the dominant gene doesn't express itself fully. So instead of the offspring looking like the parent, it begins to look, it turns out to look like something else. That's the case of incomplete dominance. So the first example I'm going to look at is the case between Mirabilis jalapa, which is also known as 4 o'clock plant. I believe by 4 o'clock, that's when it begins to close its petals. That's why it was given the name 4 o'clock plant. So this is the parent, one of the parents, which is dominant for white. This is another one, which is homozygous dominant for red. If you cross them together, white with red, the offspring will give you pink. 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 So this is what is gotten from the cross between the white parent and the red parent. Now, we're going to look at another aspect. We're going to look at probability in genetics and sex determination. In the case of probability in genetics, it has a basis with the two laws. The first law, which is law of segregation, and the second law, which is law of independent assortment. In terms of the first law, it's letting you know that no matter the number of trials, that you will end up getting the same results from latter trials. Then in the second law, it states that no matter the number of trials when two events occur together, you will end up having the same result when they occur independently. That's basically what it means. So what am I trying to say? What I'm trying to say is that, let's say for instance, let's go back to this diagram. The one of tallness and shortness. In the case of homozygous dominant gene for tallness, it will always be there when you cross two heterozygous parents together, despite the fact that the short T is there for shortness, the recessive gene for shortness is there. You will still have a tall gene out of all the offsprings. So that's basically what it means. That the crossing will involve two different types of genes, but it will still give you the same result. So even if you carry out this experiment in subsequent occasions, you will still have the same results as far as it's two heterozygous individuals that are crossed together. The same thing applicable to this. When you talk about round seeds and wrinkled seeds, the same thing occurs. But in the case of the second law, In case of the second law, if you have different types of traits, let's say for instance, you have round seeds, you have wrinkled seeds, and then you have the color yellow seeds, and then you have green seeds. Because of the presence of the color occurring together when it's being crossed with the shape of the seeds, the color does not have an effect on the shape of the seed. Neither does the shape of the seed has an effect on the color when you cross them differently. So that's basically what I'm talking about. Now, in the sex determination aspect, you know, all individuals, human beings, when you talk about mammals, we have the XX chromosome and we have the XY chromosome. Which of them is for the male? The one that is for the male is known as the XY chromosome. The one for the female is known as the XX chromosome. Now, this is how it happens. This is how the cross goes. X with X will give you offspring, XX, which is a female. X with this X will give you XX, which is another female. Y crossed with X will give you XY, which is a boy. Y crossed with X will give you XY, which is also a boy. Now we're going to look at the different characters that are involved when you talk about the sexes, the different characters that can be inherited. So we're looking at sex-linked characters. Number one is baldness. We're going to make use of Punnett square to do this crossing as well. Now, in this case, XB and XY is for the boy. This is a male. This is for the male parent. This is for the male parent. And then X capital B and X small letter b is the mother. Now, this is how it's going to be. The X raised to B is a gene that masks any characteristics relating to baldness. So this is normal. This individual here is normal. X raised to power B, Y. Y doesn't have the ability to have any character close to it. So it doesn't have any character close to it. It depends on the X for any characteristics. Whereas in the case of the female, which is the mother has the X raised to capital B, and she has the X raised to small letter b. This is the recessive trait that is responsible for baldness. But because of the presence of this capital B, it masks, it hides that expression of baldness coming from the recessive, which is the small b. So this is a normal individual with no bald head. Now we're going to cross the individual together. They get married. X raised to power B with X raised to power B, or XB rather. You have XB. This is a girl, and she doesn't have the bald characteristics. She's normal. The next child, XB and X small letter b. You have XB and X small letter b. This person is looking normal because of the presence of this dominant B, which masks or hides the small b from expressing itself. Then we'll now look at the next cross. Y with XB. XB is for normalcy. It covers, it hides the baldness. Y does not have, it cannot have any character attached to it, so it's normal. This is a normal boy, a boy with no baldness. Whereas, in this case, Y crossed with X with small b, this is a sign of baldness because there is no capital B to mask or hide the small recessive b. So the person will end up being bald. We're going to look at another one, which is hemophilia. You know, hemophilia is a very, very terrible disease because it doesn't help in terms of a situation where one is bleeding. If the person can bleed profusely, and if care is not taken, it can be an emergency situation. The person could die. So that is hemophilia. It doesn't allow the blood to clot. So it's also heritable. So let's say, for instance, this is a parent. This is one parent, and he gets married to another parent. Capital H, in this case, masks or hides the recessive, which is small h. That is the one that causes hemophilia. So look, we're looking at the first one. We're looking at the mother. The mother, which is XX, and she has the capital H and small h. The capital H masks the small h, so she's normal. Whereas, but she's a carrier. She's carrying it, but she's normal. Whereas the father has the capital H, doesn't have the small h, so he's normal. They get crossed together. So X with capital H with X with capital H will give you a girl that is normal. X with capital H with Y will give you a boy that is normal. X with small h and X with capital H will give you a normal individual, but she's a carrier because she has the gene that is responsible for hemophilia. Then X, small letter h with Y. You know, Y has no gene attached to it. It has no characteristics attached to it, rather. So X, small letter h, and Y will give you an individual that is suffering from hemophilia. So that's how it's being crossed. Now we'll now look at another one. We look at color blindness. In the case of color blindness has to do with a situation whereby an individual is unable to detect colors appropriately. He could see orange and call it red. He could see pink and call it white. So that's how bad it can be. It's an abnormality and it's also inherited. So in the case of color blindness, the one that is responsible for the color blindness, the gene that is responsible for color blindness is the small b. The small b. The capital B masks the small b. So look, we're looking at this parent. This parent is a father. He has the capital B, so that means he's normal. The mother is also normal. She has the capital B and the small b, but the capital B masks the small b. So let's do the crossing. XB with capital XB. Sorry, X capital B with X capital B will give you a normal girl. She doesn't have color blindness problem. Then X capital B with X small letter b will give you a normal individual, but she's a carrier. Then capital Y, then Y cross with X small letter b. Sorry, Y cross with X capital B will give you a normal boy. Then Y cross with X small letter b will give you somebody that is suffering from color blindness. Color blindness. But there are certain specs. There are certain specs that scientists do administer to people that have this kind of problem. There are certain specs that are being administered to these people so as to curtail the problem, so as to make them to see colors the normal way it should be. The next disease we're going to look at is Huntington's disease. In the case of Huntington's disease, the Huntington's disease has to do with a parent that has, this time around, the capital H. The capital H, anybody that has the capital H has Huntington's disease. Now, what is Huntington's disease? Huntington's disease is a neurological disorder. It affects your nervous system. It affects your movement. It affects your cognitive state. That's your mode of thinking. It doesn't make you to have a good mental state of health. That's what it does. And it can be very, very disastrous and frustrating. So this is a normal parent because it has the small h. And maybe for some case, that's the woman, for some reason, She didn't test the husband, who is a carrier of Huntington's disease. No, not even a carrier. He has Huntington's disease. One funny thing about this disease is that this disease does not surface early in life. When the person attains the age of 40 and above, that is when it begins to surface. That's one funny thing about this disease. So, the one with small h, if you cross it with the capital H, you have an offspring that is suffering from Huntington's disease. Small h with small h, this is a normal individual. Small h with capital H, this is a sufferer of Huntington's disease. Small h with small h, this is a normal individual. So we have 50% normal individuals in this cross and 50% people suffering from Huntington's disease later in life. Let's look at another cross. This is somebody that has Huntington's disease, the gene for Huntington's disease. Gets married to somebody, another individual that also has Huntington's disease. That is why it's very, very necessary to carry out tests, DNA tests, before you get into the issue of marriage. Don't be blinded by, I love you, I love him, I would die without him, I would die without her. Don't be carried away. You have to carry out this test so you can know your partner's status, so they won't affect your children. All right, so we're going to do the crossing. So capital H with capital H gives you a child that is suffering from Huntington's disease. Capital H with small h gives you a child suffering from Huntington's disease. Small h with capital H, it still gives you a child suffering from Huntington's disease. Small h with small h gives you a normal individual. So we only have one normal individual in this cross, in this Punnett square. So we have 25% of a non-Huntington disease sufferer. Then we have 75% of children in this family suffering from Huntington's disease. Now, we have come to the end of this lesson. So far in this class, we have been able to explain heredity. We have also looked at chromosome and transmission of traits, characters. We have also looked at how sex-linked characters operates. Join me as we go to the next lesson, where we'll be looking at questions and answers and how they are being answered based on this topic. Thank you very much.